Paul is an early career researcher at the University of Groningen, interested in how wild animals cope with changing environments. And so, Paul, we're ready to listen to your presentation.
Thank you. Okay. Can you see that okay? And hear me okay?
Yes.
Cool. Okay.
Yes.
So thank you very much for inviting me to speak. It's been a super interesting workshop. Before I start, I just wanted to also say a quick thank you to everyone who's contributed to the work I'm about to present. Sorry, 1 second. Yeah, it's really been very much a team effort. So as you just said, the broad focus of my research is trying to understand how wild animals cope with changing environments. And currently my main work centers around developing thermal imaging of body surface temperatures as a non-invasive way of investigating physiological state. And I'm mainly interested in finding better ways to assess physiological state because changes in physiological mechanisms are often an individual's first response to environmental challenges, and changes in physiological mechanisms drive behavior that maximizes individual capacity to survive and reproduce in the face of these challenges. And so physiological processes are likely to contain really useful information on exactly how individuals deal with the world around them. And it follows then, as we've seen in the earlier talks, that physiological parameters can also be useful as welfare indicators because they provide objective quantitative information on how individuals are coping with the conditions they find themselves in. And the specific part of my research that Wild Animal Initiative has funded sets out to exploit exactly this situation to better understand the welfare of juvenile altricial birds. So almost 90% of all bird species undergo altricial development, meaning they're pretty much helpless at hatching and rely entirely on parental care. And it's long been thought that altricial chicks should downregulate their hypothalamic pituitary adrenal or HPA axis stress response early in development. And this is mainly because they have such limited capacity to respond to stressors. And so, otherwise they could be exposed to chronic glucocorticoid exposure at a really critical developmental stage, with the possibility of lifelong negative consequences. So the evidence of this downregulation has been found in a number of wild bird species, including the white-crowned sparrow, which you can see here. But it's not clear whether the other main physiological component of the stress response, sympathetic adrenal medullary activation, is suppressed in the same way. Now, data from lab rodents suggests multiple other aspects of stress physiology do function in early development, and they are sensitive to the environment. So it's possible altricial chicks may still mount a physiological response to defend homeostasis, and that the processes involved could provide a valuable window into early life welfare. So to find out whether this is the case, we set out to leverage data collected during tests on adult birds, validating links between body surface temperatures and two sympathetic adrenal medullary associated processes, acute stress and metabolic rate. So by imaging adult house sparrows equipped with heart rate monitoring backpacks, we've been able to compare body surface temperatures with both sympathetic activation from heart rate variability and metabolic rate from heart rate to quantify their relationships. And in case you're not familiar with the concept of heart rate variability, I'll quickly explain for you. So as you can see in the electrocardiogram or ECG illustration to the right, where I've simplified and exaggerated the values just to make them clear, the time interval between successive heartbeats fluctuates. When the parasympathetic arm of the autonomic nervous system is most active, so when you're in your kind of rest and digest state, variation in interbeat durations increases, resulting in higher heart rate variability. Whereas when the sympathetic arm is dominant, so when you're acutely stressed, interbeat intervals are also much more tightly regulated, so resulting in lower heart rate variability. And this means we can use heart rate variability to determine the extent to which an individual is experiencing acute physiological stress. So for metabolic rate, Fick's equation shows heart rate is related to oxygen consumption, and so ultimately energy expenditure. And this is pretty nicely demonstrated by the impressively tight relationship between heart rate and energy expenditure we can see here from Starlings. So with a few caveats, mainly relating to the consistency of the linear relationship between heart rate and oxygen consumption, it's also possible to estimate energy expenditure from heart rate. I'm still finishing up the metabolic rate experiments, but I've just published these first acute stress results. And this plot shows the mean cardiac and surface temperature responses of 17 captive adult house sparrows to capture and handling during 38 experimental runs. In the first column, we have the mean baseline values measured for each trait immediately before the experimenter entered the bird room. And then in the second column, we have the mean continuous response during the 3 minutes that the experimenter was in the bird room, during which they caught and then released the bird. And then in the third and fourth columns, we've got the mean values for each trait measured at 15 and 30 minutes after the start of the stressor. And as you can see, heart rate jumps by about 300 beats per minute as soon as the experimenter enters the room and stays elevated right through the stressor period and returns to baseline levels by 15 minutes. And this increase in heart rate reflects sympathetic activation with heart rate variability dropping immediately and then remaining below baseline for the entire stressor period. Remember, lower heart rate variability means greater sympathetic activation. And then again, it returns close to baseline by 15 minutes after the stressor started. So clearly capture and handling activated the sympathetic acute stress response. And if we compare this with the surface temperatures, here the eye region and bill, we can see both drop below baseline during sympathetic activation, especially in the bill, and both return to more or less baseline when the sympathetic nervous system does the same. And happily, this confirms surface temperatures track sympathetic activation and that we can use body surface temperatures measured non-invasively by thermal imaging as a proxy for sympathetic activation during acute stress. And armed with this data and the data that we expect from our metabolic rate tests, we should be able to use thermal imaging to work out whether altricial chicks do indeed mount sympathetic or energetic responses in early life despite downregulation of the HPA axis. So that's exactly what we've been doing. So our specific aim was to compare patterns of surface temperature change measured from developing chicks during stress and energetic manipulations with those that we'd validated against sympathetic activity or metabolic rate under similar circumstances in the adults. And if juvenile surface temperature dynamics are similar to the adults, then we can reasonably expect that the same processes apply. And if not, this would instead be a first step towards demonstrating downregulation of the sympathetic stress response as well as the HPA axis in altricial juveniles. So it's a useful outcome either way. So unfortunately, our house sparrow breeding program didn't quite work out. So we switched to zebra finches for the juvenile work, which are considerably easier to get to breed. And given the physiological processes we're targeting are so highly conserved amongst endotherms, it's quite unlikely that switching between relatively closely related species represents a major issue. So once we decided on the species, we needed to develop a way of imaging within the nest boxes the birds used to breed, and this basically involved designing a kind of lid to support the thermal camera at its minimum focus distance, so to make the chicks as big as possible in the field of view, with enough flexibility to accommodate the kind of natural variation in nest height. And we also added a visible light video camera to be able to identify individual chicks from color marks, because these wouldn't be visible on thermal video, and an LED light for the video camera. And the whole thing was run from a single Raspberry Pi per nest box using sort of bespoke Python code, and it was all managed over Wi-Fi, so it was really convenient. And yeah, this is what the footage looked like. So in terms of the treatments, we gave the chicks 6 days of alternating randomized acute stressors or food restriction between the ages of 5 to 10 days post-hatching. And this is the approximate period over which the chicks transition from ectothermy to endothermy, because they can't generate enough heat to maintain their body temperature when they've just hatched. And so this is the period where we'd expect the biggest changes in surface temperature responses. So on acute stress days, there were 4 randomly timed and ordered 30-minute periods of stressors, 1 each of cage tapping, which is essentially running a pen along the cage bars to make a noise and vibration, which we also did with the adults, so we could kind of directly compare those stressors. Rolling the cage rack backwards and forwards, human voice and presence, and radio white noise, all of which have been shown previously in the lab I'm working in to trigger a HPA response in adult birds. So we kind of hope that they're all stressors. And the parents were blocked from entering the nest box during the application of the stressors, so the chicks would remain visible to the camera throughout. For the food restriction, we decided on 4 60-minute periods per day where we'd block parental access to the chicks to prevent them being fed, rather than 30 minutes to match the stressors. And this is because 30 minutes of food restriction probably wouldn't generate a strong enough effect, because it's probably too close to the normal gaps in provisioning that we'd see in zebra finches. So we started the cameras recording 10 minutes before each treatment to establish a kind of baseline, and then the camera was left running for the duration of the treatment, generating about 7.5 TIFF images a second, which we save for later analysis using the open source image processing package Fiji. And we're still very much in the process of extracting the chick surface temperatures, which basically involves drawing around the target body region in Fiji and then extracting temperature data from that part of the image. It's probably a bit difficult to make out, but you can probably see here that we targeted the whole head in the juveniles rather than the bill or the eye region that we used in adults, because both are too small in the early stages to be reliably separated from the background and the rest of the body. But anyway, because we are still extracting the temperatures, I'm afraid, yeah, I don't have data to show just yet. But if we're going to treat the project as a case study for validating potential new welfare measures, what we can say at this point is that by recreating the design of our adult experiments as closely as possible for the chicks, including kind of measurement techniques, treatments, and sample sizes, we've really capitalized on the adult data, achieving what is probably as close as possible to a true validation without directly measuring sympathetic activation or metabolic rate in the chicks. And by taking the opportunity to take many more repeated measurements than we could have done with the instrumented adults, we've also managed to tailor the project to the chicks' specific circumstances. So that allows us to collect really valuable information on how thermal responses might change during the development of endothermy. So although it's definitely correct to say that a full validation, so in our case comparing chick surface temperatures with direct measurements of sympathetic activation or metabolic rate, would usually be ideal, I think, as Melissa points out, I think it was her method 5, it is sometimes possible to generate kind of similarly instructive data on potential new welfare measures with less effort and potentially less ethical issues through strategic use of existing data. And that's it. Thank you. Thanks for your attention.